Hello children, in the last class we have studied about principle of inheritance. While studying the principle of inheritance, we came across with various types of crosses, that is monohybrid cross and dihybrid cross. Let us see the monohybrid cross once again. Here you can see that the parents, two contrasting traits are taken and it is crossed. Now you see the F1 generation. F1 generation, all the progeny shows only one character, that is the tall character. Phenotypically this plant is tall, genotypically they are heterozygous dominant. Now come to the F2 generation ratio of F2 generation, that is phenotypic ratio, that is 3:1. So that means 3 tall plant and 1 dwarf plant. Phenotypically, that is, what is phenotype? That is the physical appearance of the plant. Phenotypically the plant appears tall, but genotypically this plant is homozygous and these two are heterozygous. So, and now come to this plant. It is a dwarf plant which is a recessive trait of height. The recessive trait always appears in the homozygous condition. So after seeing a dwarf plant or recessive plant, it is very clear that it is having a homozygous, that means the gene is in homozygous condition. But this plant, we can't see physically whether it is a homozygous or heterozygous. So we can call this plant as unknown dominant. So in order to find out the genotype of an unknown dominant, there is a cross that is known as test cross. So test cross. So what is a test cross? Test cross is done in order to find out the genotype of the unknown dominant. Phenotypically all the plant appears tall, but genotype varies. It may be homozygous or it may be heterozygous. Okay, so a test cross is done in order to find out the genotype of the genotype of that unknown dominant. Now you see here, we have taken a homozygous tall plant. If it is crossed with dwarf variety, what is a test cross? Actually, a test cross is done by crossing the unknown dominant with the recessive trait. Crossing an unknown dominant with the recessive trait. So here this is an unknown dominant, that is homozygous tall, is crossed with recessive trait. In the F1 progeny, all the plant shows only one character, that is all the plants are tall. Okay, 100% result we are getting. Okay, now come to the next cross. This is a heterozygous tall plant. When it is crossed with a recessive trait, that is dwarf, in the F1 progeny, 50% of the progeny shows tall trait and 50% of the progeny shows dwarf trait. What you have learned from this cross? That means if an unknown dominant is crossed with a recessive trait, if all the progeny shows only one trait, that is a dominant trait, we can say that the unknown dominant is homozygous. Okay. Now next, if an unknown dominant crossed with recessive trait, 50% of the progeny shows tall trait and 50% of the progeny shows dwarf trait, we can say that the plant or the unknown dominant is heterozygous. Is it clear? So that is known by the name test cross. What is a test cross? Test cross is done in order to find out the genotype of the unknown dominant. Okay, how it is done? It is done by crossing the unknown dominant with recessive trait. Okay, so if all the progenies are showing only one trait, we can say that the parent is homozygous. And if you are getting the result 50-50, 50 tall, 50% plants are showing tall character and 50% plants are showing dwarf character, then the parental plant, that is unknown dominant, is heterozygous. Is it clear, student? Now, Mendels experiment on P was also done in other plants. Okay, so like the same methods are used. What are the 3 steps of Mendels experiment? That is selection of tall, selection of pure parent plant, okay, hybridization of the pure parent plant, and self-pollination in the hybrids. These are the 3 steps followed by the Mendel. So, these 3 steps are followed in other plant also. So, Antherhinum, majus and Mirabilis jalapa. This plant is having 2 contrasting traits of flower color. Suppose we are taking the parents, it is having red color flower. When it is crossed with, its contrasting trait is white. So, its genotype is capital R, phenotype is the phenotype is red and genotype is capital R, genotype is small r, phenotype is white. Okay. Now, see the gamete. Gametes. It produces gamete, that means it followed the principle of segregation. Alleles of the gene separates and the gamete receive only 1 allele, and here also gamete receives only 1 allele. In the F1 generation, so when it is fused, capital R, small r is formed. According to principle of dominance, so if the plant is followed the principle of dominance, what would be the color here? Here it would be red color. Okay, but the flower produced a different color which is known as the pink color. This flower the plant produced a pink color. Okay, so that red color is not appeared. So what you have learned from here? That means the principle of dominance is deviated here. It is not followed the principle of dominance. There is a deviation from the principle of dominance. Again, this plant is self-pollinated. So it produces both the male gamete and female gamete. We'll write here as gametes. Okay, here the female gamete, F2 generation. We'll see what is the F2 generation. You can make the Punnett square. That is, suppose it is a male and here we are taking female, capital R, small r gametes, capital R, small r. This gamete fuses with the other gamete to form capital R. Here it fuses to form capital R, small r. Here it fuses to form capital R, small r. Here it fuses to form small r, small r. This is the possible genotype. Now, what is the phenotype? That is red, pink, and white. So let us write the phenotypic ratio. Phenotypic ratio is 1 red, 2 pink and 1 white. Is it clear? Now, genotypic ratio. Genotypic ratio, that is 1 red, that is homozygous dominant, 1 heterozygous dominant, 2, 1 and 1 homozygous recessive. Again, the phenotypic ratio is also altered. In pea plant, what we have seen, the phenotypic ratio is 3:1. Here the phenotypic ratio is 1:2:1. That means phenotypic and the genotypic ratios are same. So what we have seen here, this allele is unable to dominate completely over the other allele. Okay, what we have studied in monohybrid cross, one allele completely dominates over the other allele, so we are calling it as a dominant allele. Here this allele capital R is unable to dominate completely over the other allele. Okay, so this type of inheritance is known as incomplete dominance. incomplete dominance. Okay, so this allele is unable to dominate over the other allele completely. That is why an intermediate color is formed, pink. Okay, so this type of inheritance is known as incomplete dominance. So you'll get questions like name the inheritance where both the phenotypic and genotypic ratios are same. That is 1:2:1 phenotypic ratio and genotypic ratio is also 1:2:1. The inheritance is incomplete dominance. Is it clear? Children, till now we have studied that one gene carries two alleles. One gene carries two alleles, like one dominant allele and one recessive allele. Okay, one gene is carrying two alleles, that is one dominant allele and one recessive allele, like tall plant is a dominant trait and dwarf plant is a recessive trait. But there are certain genes which is having more than two alleles. Such alleles are known as multiple alleles. Multiple alleles. That means a gene is having more than two alleles are known as multiple allele. Example of multiple allele is blood group in human. That is ABO blood group. Okay. Now, blood consists of plasma and blood cells. What are the blood cells? Red blood cells, white blood cells and blood platelets. Now, how the blood groups are formed? Now, you see here, there are how many types of blood groups are present? A, B, AB and O. This is the blood groups. This is the blood group. Okay. Now this A blood group is due to antigen which is present on the plasma membrane of the RBC. RBC is having an antigen on its plasma membrane. Suppose these are the antigen which is present on the plasma membrane. This antigen is A antigen. A blood group is having antigen A. B blood group is having antigen B. Antigen B. Suppose this is antigen B. And AB is having both A antigen as well as B antigen. You can draw like this. B antigen. It is having both the antigen. Suppose this is antigen A and this is antigen B. Due to the presence of both the antigen, this blood group is showing AB. And in O, RBC do not have any antigen. Okay, RBC do not have any antigen. So now, this antigen is made up of a sugar polymer. Antigen is made up of, we can say it is an antigen which is made up of a sugar polymer. This antigen is made up of sugar polymer. There is a gene which is known as the capital I. This gene is responsible for the synthesis of sugar polymer. Okay, and this gene occur in, it is having three alleles, capital IA, capital IB and I0. These two, capital IA and capital IB, are dominant, and this is recessive, that is capital I0 or IO is recessive. Children, you know that human beings are diploid. They are having, each human beings are having only 2 alleles of this blood group. Okay, let us see the genotype. We consider this blood group as phenotype. Phenotype means that is the physical appearance. How? By testing a blood we can find out the blood group. So we consider it as a phenotype. Now, what is the genotype? If a person is having A blood group, there is 2 options. It may be in homozygous condition, the alleles may be in homozygous condition or in heterozygous condition. This is heterozygous condition, one allele is dominating over the other, so it is followed the principle of dominance. This is completely dominating over the other allele. So this type of alleles, this is homozygous and this is heterozygous, and it forms the blood group A. Similarly, here you can see IB is one option, and there is heterozygous IB, I0. It is homozygous alleles, it is heterozygous alleles. So in this form, in this if the gene is present in this form also, this blood group is formed. For AB, capital IA, capital IB, there is no other option. That means these 2 alleles are dominant alleles. These 2 can equally express, that means they togetherly express its character. None of them are dominating over the other. Okay, so A can able to express its character and B is able to express its character. That is why they are forming a AB blood group. Okay, so what we can say here, as they can expressing togetherly, we call it as a co-dominant. This is a co-dominant gene. Why it is called co-dominant? Because they can able to express togetherly. So again it is deviated from the principle of dominance. Okay, both the character of a alleles expresses togetherly, so we call it as a co-dominant gene or co-dominance. Now here O is a recessive trait. It can express only in the homozygous condition. So what we have seen, how many phenotype is there? There are 4 phenotypes. There are 4 phenotypes, and how many genotypes are there? There are 6 genotypes. 1, 2, 3, 4, 5, 6. 6 genotype and 4 phenotypes are there. Now we can see a cross. Let us see a cross. Suppose a father is having blood group A. Mother, she is having a blood group B. Both the father and mothers are having, their alleles are in heterozygous condition. The genotype of this father and this is the phenotype of mother. That means the alleles are in heterozygous condition. Now gamete, how the gamete is formed? Alleles get separated. So these are the gametes of father and this the gametes of mother. Alleles get separated during gamete formation. Okay, so we'll make the Punnett square here. Fathers and mother. IA, one gamete, I0, another gamete here. Mother's is IB, one gamete, other is I0. So it fuses to form IA, IB. Here it fuses to form IB, I0. Here it fuses to form IA, I0, and here it fuses to form I0, I0. What is this is the genotype of the blood group, and this is the phenotype, AB blood group. This shows B blood group, and this shows A blood group, and it shows O blood group. What you have seen, a parent who is having A blood group is crossed with mother who is having a B blood group. There is a possibility of appearance of all the 4 blood groups in their progeny. The new blood groups are AB and O. Is it clear? So this is about the multiple allele. What is multiple allele? More than 2 alleles are present for a gene. So what is a codominance? Codominance means both the alleles are able to express togetherly. That is known as the codominance. Now, up till now what we have studied that one gene is responsible for one character. Okay, one gene is responsible for one character. There is a gene which is known as pleiotropy. Pleiotropic. In pleiotropic gene, one gene is responsible for more than one trait, more than one trait. Till now we have studied one gene is responsible for one character. Here, this pleiotropy gene, one gene gives more than one trait. Okay, trait means character. Okay, example is sickle cell anemia. Sickle cell anemia is the example. Here you can see that this disease is due to a mutation which takes place in the DNA. That mutation is known as point mutation. Okay? Due to point mutation, there is a genetic disorder. That genetic disorder is known as the sickle cell anemia. These patients suffer severe anemic. They are severe anemic patients. Due to this point mutation, there is a change in the structure of the hemoglobin. Where the hemoglobin is present? Hemoglobin is present in the RBC, red blood cell. Okay, inside the red blood cell you can see the hemoglobin. This is iron part of the hemoglobin, and this is the protein part of the hemoglobin. Okay, this globin is made up of 2 alpha polypeptide chain and 2 beta polypeptide chain. So mutation takes place in the 6th amino acid of the beta chain. So you will see this is the beta chain. 1, 2, 3, 4, 5, 6. Actually, the amino acids are here. Valine is present. Here, histidine is present. Here you can see leucine. Here you can see threonine. And here you can see proline. And here, glutamic acid. For a normal case. In a normal case, glutamic acid is present. But due to point mutation takes place in the DNA. Where the point mutation takes place? Point mutation takes place in the nitrogen base of DNA, due to which glutamic acid is changed into valine. So the structure is changed. Structure of the hemoglobin is changed. As the structure of the hemoglobin is changed, the shape of the RBC is changed into sickle shaped. The RBC is changed. Now due to the change in the RBC, the content of the hemoglobin is also reduced. So the carrying capacity of oxygen is reduced. Okay, so that causes anemia. And moreover, the shape of the RBC blocks the flow of blood. Okay, so what you have seen, these are the problem which is caused by the structure of the hemoglobin changed and shape of the RBC is changed. But these patients can survive in the malaria prone area. Why? Because The malaria parasite can survive in the RBC. Okay. So, for the multiplication of malarian parasite, what they require? They require RBC. As the shape of the RBC is changed, what happened? This parasite is unable to survive here. Okay. So, the malarial parasite will not develop in their sickle cell anemic patients. They are not prone to malaria. They can survive in the malaria prone area. Okay. So, what you have seen that pleiotropy means one gene is responsible for many traits. So, what is the trait here? Change in the structure of the hemoglobin causes the change in the shape of the hemoglobin that caused anemia. Okay. But this patient can survive in the malaria prone area because parasite is unable to survive in the RBC. Okay. So, they can live in the, or they can survive, they are resistant to malaria. Is it clear, children? Now, in the next class we will study other types of inheritance. Thank you.